Good evening, everyone, and warmest welcome to tonight's panel discussion about Alzheimer's and Alzheimer's-related diseases. This evening is hosted by Johannes Gutenberg University Mainz, the Stifterverband, and the German Center for Research and Innovation. My name is Dietrich Wolf-Henner, and since October 1st, I'm the program manager for the German Center for Research and Innovation here in New York. Tonight, we are very happy to co-host this event on the important discussion about new targets to prevent and to treat Alzheimer's disease. The German Center for Research and Innovation New York was established in 2010 to showcase Germany as a land of research, science, and innovation. We provide a platform for leaders in science, technology, and the humanities to engage in transatlantic exchange and collaboration. The German Center for Research and Innovation New York is one of 5 centers worldwide that help build relationships between our host countries and the German science and innovation communities. We are active in Russia, Brazil, India, Japan, and of course, here in the US. We foster discussions about the current innovations such as AI, blockchain, or precision medicine and their benefit for people and society. Today's event is a perfect example for how we empower the dialogue between the public, science community, policymakers, and industry. And the timing is perfect too. Surprising data from a potential new treatment option for Alzheimer's has made a lot of news this week. And the amount of reporting showed me again what is at stake. Millions of people waiting desperately for a breakthrough. The risk of failure is high, and the business opportunity is enormous. Alzheimer is a frightening disease, and it's personal. I saw personalities of many people vanishing when I was working in a nursing home. And on a private note, I had to witness the progression of the disease with a family member. My husband's grandmother lined up on our wedding reception 4 times to give her blessing and congratulation. That was heartwarming and cute in a way, but also alarming. 2 years later, she didn't know our names. And then she lost her language. She became more and more helpless and lost. In the months before she passed away, her own anger about her state was the most terrible thing to witness. And I'm sure all of you know someone and know these stories about someone who had Alzheimer's or a related disease or who is still fighting Alzheimer. So with these stories in mind, I want to hand over the evening to our scientists tonight to get inspired and to hear about potential breakthroughs that are on the horizon. So thank you all for joining us tonight, and I have the pleasure to hand over the mic and the discussion to Fazi Bekulat. Thank you.
Good evening, everyone, and welcome to this evening. I have the pleasure to introduce this evening's panelists. These are Professor Ralph Nixon from NYU. He is also the director of the Center for Dementia Research at the Nathan Klein Institute. Professor Christian Beyer from the Institute of Pathobiochemistry of the University Medical Center of the Johannes Gutenberg University in Mainz, Germany. We have Dr. Mariel Owen from the Alzheimer's Drug Discovery Foundation, and also Dr. Markus Nielbock from Hoffnungsbaum e.V. So we came here together for a day workshop on Alzheimer's-related disorders in autophagy, and we connected and discussed and hopefully had great impulses we would like to share this evening with you. And we all did this in New York, and if you ask why New York, because, well, it's New York, but so it's kind of obvious. But New York has a bit of a history, so research of autophagy in the context of neurodegenerative disease actually started out kind of here with Professor Nixon in the forefront of this research, for example, and there are a lot of brilliant brains working on autophagy in the context of neurodegeneration in this area, so this area is well connected. So, well, New York is not only beautiful, it's vibrant, it's connected, and it's very, very busy. So, and one thing that strikes you, what struck me every time I come here, well, it's my second time, but well, is the traffic. There is a lot of traffic in Manhattan, especially. And I'm in awe of everyone who comes here and knows how to get from A to B on time, actually, because I think it's very, very exhausting over time. It needs a lot of planning ahead, and it needs a lot of energy, and I think it's over time quite, well, exhausting. And ideally, in a city like this, everything would be regulated and would work well, and then you would get from A to B whenever you want. But there are actually good examples of great cities who know how to regulate their traffic, their infrastructure, everything like this. And that's your average cell. Every cell in your body is like a city. It's just as crowded as New York, maybe even more, but it knows how to regulate its manpower, its infrastructure, everything actually. So depending on the area where your cell or your city is located, like a state, it may have different regulations, but it will solve its problems just as effectively. So, for example, if you have a construction site, well, it will be there just for a short amount of time, and then the thing will be standing there just as ready. And so you have a factory building, for example, that is not used anymore, so we're going to tear it down, we can use that space and recycle the material and use it. If we have something in the streets lying around, garbage accumulating, we send the garbage disposal team, recycling team, and they will get rid of it before it can accumulate and get toxic. So, but over time, imbalances can occur. So infrastructure over time gets used up, and also it's important to see that imbalances can affect your infrastructure badly. And depending on the area where your cell or your city is located, over time, these imbalances can lead to impairments. But importantly, depending on where it is, it can be maybe compensated, maybe not. But one area that is very, very vulnerable to imbalances and impacts from the outside is the area of your government. So if imbalances happen here over time, this can impact your whole body. Just as a government that doesn't work well impacts the whole country, this is the same in our bodies. So I have the pleasure to invite all our panelists here to put into context Alzheimer's, related disorders, and autophagy, to put into perspective in the picture I just painted here. Yeah, we will start with the talks first. We will have, okay, you will, okay. So we start with Professor Bale, who will give some insights into autophagy.
Yeah, the analogy was quite good about the traffic and the traffic jam and the control of things in cities or in the cell. And to build the bridge to that, what we have heard in the introduction of Dr. Fenner actually is, yes, we are talking about Alzheimer's disease, but you also said there is, we don't have a cure yet, and so we are searching for cure. And so we are searching also for new mechanisms. And this was actually the purpose of our scientific workshop meeting, to sort of discuss and to address the concept of autophagy. How is it linked to neurodegeneration, including Alzheimer's disease? And what can we learn from it? Is it perhaps a new way to think about the disease or to have also a new basis of therapy. That's the reason why I'm starting with a short presentation, just to introduce what autophagy actually is in a few slides. Now, inside the cell, and this comes back to the picture given by Fasi actually, it's crowded. What you see here is these are basically proteins. So the proteins in your cell. So the cell is stuffed full of proteins, and we call it protein crowding. And you also have to know that every protein has a function in your cell. So it is architecture protein, it is an enzyme, it is, taking care of so many different functions. But in order that a protein can function properly, it needs to have the right three-dimensional conformation. The figure must be right. And this we call the folding, the correct folding of proteins, just as symbolized here by this paperwork. You need a correct folding for a correct function, and if there is misfolding, these proteins cannot function. They just hang around in the cell, and as the garbage in the street, if it's not pulled away, it's accumulating and then disturbing function. And so every cell of the body, including in particular the nerve cells of the brain, they need a proper system to control the generation of proteins, the folding of proteins, and if proteins are misfolded, also the removal of proteins before they really accumulate in the cell. This is symbolizing two main pathways that we have in our cells that take care of misfolded proteins. One is the so-called proteasome, which is a multi-enzyme complex and attracts proteins that are, every protein has a half-time of survival, so to say, and then it becomes dysfunctional. So after this time, there's a physiological process to remove this protein by digestion in the proteasome. And then you can sort of take the pieces and use this for recycling and build up new proteins. But this process, although running every second in our cells, has limitations. And the limitations are if these proteins are too large or too clumpy and too big aggregates, it is no longer functioning, the degradation in the proteasome. And for this process, for this problem, we need an additional process, and this is the autophagy, the process called autophagy. And if you had Greek in your school time, you realize that autophagy means self-digestion, meaning that proteins, but not only proteins, but also pieces of your organelles in the cell, mitochondria, pieces of different vesicles, whatever is hanging around, having no function, it is engulfed by membranes and then fusing with compartments in the cell, the so-called lysosome, which then degrades it very effectively. And this is also a process of recycling then. So you have pieces and building blocks for new cell divisions, for new protein buildup and so forth. So these are the two basic mechanisms. And our workshop today was dealing mainly with this autophagy process because, as we will hear from Professor Nixon in the following short presentation, there is for quite some time a good indication that there is a disturbance of this process during Alzheimer's disease. There's clear proof that this is going on, and we as scientists, we need to work on this and then try to find ways to sort of perhaps use this as a potential target. Now, what's the control of autophagy? I can show you this slide, and this looks complicated, and it is complicated. And it's just showing that these proteins all are involved in autophagy control. So, and it's far too much information to explain it in a few words, but I can try it with another more simplified slide, which brings us back to the origins of autophagy research, which was driven by Yoshinori Osumi. He received the Nobel Prize for Medicine or Physiology in the year 2016, actually, for his groundbreaking work, how autophagy is controlled. And he worked with yeast, not with human cells or animal cells. He worked with yeast, basic yeast cells. And what he found is that whenever you give the cells a hard time, you say we starve the cells, then they need energy, then they need building blocks, and then they sort of start to digest their own proteins in the cell. So again, creating building blocks and energy for function and viability. And this is then like, you know, your trash that you are, in addition, the trash that you are accumulating is all digested through this autophagy and then finally fully recycled. And this is in a more biochemical way drawn like this. Again, you see it starts here with membranes. These are just lipids that form little blebs in the cells. They can engulf these trash that needs to be degraded and cleared. And then this engulfment closes up, fuses with the lysosome, and it's degraded, and then this cycle is going on and on. When the cell is really in bad shape, this is really induced by starvation, but this is a process that is also continuously going on in the cell, and also in particular in nerve cells. So, and the process we are very much interested in is, you know, for a long time it was believed that this autophagy process is rather unregulated, and it's happening or it's not happening, you know, there's no real control. But based on the research of the last 10 to 15 years, actually, we realize that this is a very selective process. And just like if you sort of sort your kind of trash, we do this in Germany very, very well, the cell has proteins that recognizes particular junk that needs to be degraded, and this is sort of the selectivity of the process. It's not randomly occurring, it's really selective process, and the basis, the biochemical basis of this selectivity are particular proteins and so-called receptors. Now, and for all different types of degradations in the cell, we have different types of selective autophagy pathways. And this is just symbolized, so you collect your paperwork and put it in particular trash cans or your plastic or whatsoever. And this is things that are getting into the cells, like microbes and viruses can be degraded by autophagy. Defective mitochondria can be degraded by autophagy, and so forth. So, and of course also proteins that are then, before they aggregate or when they are aggregated, they are also degraded by autophagy. Just to show that it's a complex network of regulations, and this particular network is disturbed in Alzheimer's disease. And this process, since it's such a basic process occurring at every second in our cells, is implicated in many different disorders, including neurodegeneration, but also infectious disease, vascular disease, cancer, and metabolic disease or pulmonary disease. So it's such a basic process that in all these different types of disorders, you have to realize that there are disturbances. In many cases, we don't know what is cause or effect, if autophagy disturbance is the cause of a disease or is just a consequence of the disease. So finally, and this is based on the fact that we on the panel have a representative of a particular group of autophagy-caused diseases, I would like to shortly introduce that all these proteins that are involved in the control of autophagy, some of them can be mutated, and there is then leading directly to disease. And this is called a monogenetic disease. Yeah? If one gene is mutated and then causes directly a disease. And this is the case for a protein called YPI4, which is involved in this building up, in the build-up of these first membranes here. And if there is a mutation, the consequence is a neurodegeneration, a developmental disease affecting children called BPAN. And as I said, we have a representative of a foundation, and he will be also here and will explain on this disease. So finally, I would like to summarize with this one sentence that autophagy is a cellular recycling process with implications for many human diseases, including Alzheimer's disease. And with this, I would like to stop here and hand over to Professor Nixon for a short introduction on Alzheimer's disease.
Well, thank you, Christian. I think that was excellent. The process of autophagy is really complicated, and I really congratulate Christian for, as complex as it might have sounded to you, this was a really a very clear overview of the process. So what I wanted to do, since the focus of this meeting was Alzheimer's and autophagy, is to provide the context of Alzheimer's disease. I don't know the background of the audience, but I'm guessing that some of you, the prevalence is so high that some of you have probably experienced knowing someone with the disease or having a close relative with the disease, and you know what a devastating disease it is. So I'm going to just go simply through a few facts about the disease and try to tie in a few of the things that Christian was mentioning. So Alzheimer's disease is the most common form of dementia, and a lot of people equate dementia and Alzheimer's. Dementia is actually a set of symptoms, the core one being a memory impairment, but then to really be qualified as dementia, one has to have at least one of these problems: difficulty expressing yourself, recognizing people or objects, performing complex motor activities like cooking or even dressing at some point, or these more complex tasks like planning your day or doing abstract kind of thinking about a subject. And there are now at least 44 million people with this, with either dementia in general or Alzheimer's disease. So this is the most prevalent form of dementia in the world, and it affects all populations relatively equally. There are very limited evidence that a particular country has lower prevalence unless the people have an average lifespan that's very young and they die before they get the disease. These are some of the other forms of dementia, and you'll recognize most of them This FTD is frontotemporal dementia, which is a disease very commonly misdiagnosed as Alzheimer's in the past, but now we have diagnostic criteria to differentiate them. DLB is diffuse Lewy body disease, which is a hybrid between Parkinson's and Alzheimer's, where you have symptoms of both, and it's quite devastating. Alzheimer's, like many of the other conditions that I just showed, are diseases of aging, and none more so than Alzheimer, that the prevalence exponentially increases as one gets older, to the extent that by the age of 85, there's as many as 50% of individuals have a degree of dementia. This doesn't mean a degree that would be a devastating effect on the family or lifestyle, but enough to have the diagnosis that would progress over usually a period of 6 to 8 years to a worse condition that is essentially debilitating. So this is who the disease was named after. This dapper guy is Alzheimer, and next to him is Emil Kraepelin in Munich. They're sitting around. Emil Kraepelin was the psychiatrist who was trying to differentiate these puzzling forms of psychiatric disease, and to do it by looking at the brain and staining it with various types of stains to reveal pathology. And Alzheimer had a special stain that he had developed, a silver stain. And at the same time, he was seeing some of these individuals, and this woman was a patient who presented with some memory disorder, some confusion that was relatively mild at the time, but over a period of 6 or 7 years resulted in her basically being bedridden. And she died with very severe memory loss, as well as some of the other symptoms of dementia that I mentioned. And when he looked at the brain, with the silver stain that he had, he found these two unusual pathologies here. One was called the neurofibrillary tangle, NFT, which is inside the brain cells, and it's an accumulation and aggregation of proteins similar to what Christian was talking about, but it's a very specific protein, tau, which some of you might have heard about. The other lesion is this senile plaque or neuritic plaque, where these degenerating structures that are coming out from the neurons are intermixed with this brown substance, which later was found to be amyloid. And amyloid, as I think Christian mentioned, it's been the focus of most of the therapies, the target of most of the therapies over the past 15 or 20 years. You can see the shrinkage of the brain because what happens in addition to these lesions is the massive loss of brain cells that occurs. In addition, though, and what is the subject of today's discussions, were that here's a plaque, again, stained this time with an antibody to amyloid to recognize specifically the amyloid component, and also to stain with something that recognizes this tau protein that accumulates in these tangles, which you can see here. And these little curly cues all over the place are processes that usually communicate from one cell to another and allow the messages to be passed from one circuit in the brain to another, which allows you to think and to do all the things that you need to do. These, it turns out, if Alzheimer had had an electron microscope, which he didn't have at the time, and which we had, you find that these little structures that are the bridges between cells are filled with this material. And what this is basically metabolic waste. It's the equivalent of these structures that Christian had shown you earlier in different forms. So, and here is a similar visualization in the cell body. This is as the cells become more compromised, they fill up with, so the brain is literally filled with waste, and this makes Alzheimer's different than other diseases in the profundity of waste accumulation that occurs in the brain, which is why we started looking at autophagy originally. And this is the most complex of the slides that I'll show, but it's to tie together what Christian was mentioning, the autophagy pathway, where materials inside the cell are sequestered in these compartments and then normally would be degraded within these recycling compartments called lysosomes. In Alzheimer's disease, these lysosomes fail. And what happens is the compartments that contain all of that junk that I just mentioned start to accumulate because there's no way to get rid of them. And in addition, there are other pathways that are bringing in things from outside a given cell and carrying them to the lysosome. So the lysosome is getting fed this excessive amount of material that should normally be degraded that's not being degraded. And it's made worse by aging, which also corrupts this process of recycling. So what's depicted here as a single dystrophic neurite at a very low power in the brain, this is a much lower magnification, there are hundreds of these structures in this, this senile plaque. And if you look around, you see that there are hundreds of these plaques. So you can multiply, do the math, and you realize that the brain is chock-a-block full of material that's not being cleared. Something that you may or may not know is that by the time you develop even mild cognitive problems, these pathologies have already shown up 5 to 10 years earlier, and even the biology that I described that starts with this inability to clear material from autophagy is starting even as much as 20 years before symptoms are occurring. And so by the time you actually develop what could be diagnosed as dementia, you already have a very advanced pathologic situation. And this has been given as one of the reasons why the drugs that have been applied at this stage in the disease, which is most of the clinical trials, are trying to stop a train after it has reached maximum speed. It's just a lot harder than if you put a little block in front of the wheel in the train station and prevent it from starting. So all of the treatments now are trying to be aimed at this earlier stage, and ideally what we were talking about today is to interrupt the disease at its origin, which some of us, including myself in Alzheimer's disease, think it's this corruption of this clearance mechanism, which allows these other proteins that are more well known, the amyloid and the tau, to accumulate because they're normally cleared in the brain through this autophagy system. So everything can kind of be traced back to this very early biology, and if you could somehow intervene and start to get that system working more effectively, you could, in theory, prevent all of the clinical symptoms of Alzheimer's. So that's the goal with Alzheimer's, a similar goal with many of the other Parkinson's and Lewy body disease. They all have the equivalent of this tangles and plaques, different proteins, different look to the lesions, but same philosophy here. So there is a goal toward being able to at least detect this stage of amyloid, which is well before clinical symptoms. And this is done by using PET scanning, which can detect, even in these mild cognitively impaired individuals, this growing amount of amyloid that's shown in red here. It should be mentioned that this technique is so sensitive that even people who are cognitively normal can have quite a bit of amyloid in their brains, and the presumption is that they would go on and develop Alzheimer's, but it's really not been nailed down that everyone who has amyloid in their brain is going to get Alzheimer's disease, and that there's this concept of functional reserve, which is a very vague term for the resilience to pathology that people have. And the strategy, which includes enhancing autophagy, is to build up this resilience to even something that might be building up as a toxic process in the brain, to resist it and to prevent cognitive. So there are different ways to look at how autophagy might be involved in clearing this junk out or making the brain cells more resilient to being impacted by it. So the approaches that have been tried have mainly been, as Christian mentioned, this amyloid vaccine or anti-amyloid treatments, some beginning to target tau, and they haven't been working as we had hoped. And there was someone referred to a study this week that came out that suggested perhaps a little glimmer of hope with one of the vaccines that seemed to be showing some benefit. But this is far from, papers tend to hype these discoveries, as you probably know. And there's a lot more work that needs to be done. The current treatments are ones that try to enhance the communication between still surviving connections between one neuron and another, and these are the drugs like Exelon and Aricept that are enhance these chemical communications, and this drug memantine, which is a drug that stimulates the response to these various chemicals. Those are not drugs that will delay the disease. They treat symptomatically. The disease still progresses, but does have some benefit. So, like I said, and Christian said, at least 80 to 90% of the research that's been done so far has been done targeting mostly amyloid and more recently tau. Almost none of the research has really seriously approached this other arm that would lead to survival and resilience of the brain to a disease like Alzheimer's or Parkinson's or any other late onset disease. This is where autophagy falls into that category because it is it's both protecting by clearing toxic materials, it's repairing by recycling things that are necessary to regenerate structures within the cells that are, and if you can't recycle, you don't have the building blocks to do this. So this is one of a number of avenues that are being explored, but one that we think is very promising. And this is, without getting into the weeds about this, this is actually even an older slide, but these are different drugs and different genetic ways to manipulate the autophagy system that in mouse models of Alzheimer's disease and even in mouse models of Parkinson's, these drugs that enhance the ability to clear proteins through this pathway have remarkable therapeutic effects. And you've heard this with amyloid vaccines too, which cure mice left and right, but don't work in the clinic. There are different reasons why that's the case with amyloid drugs. I think the promise here is that this is a system that, if it were manipulated properly, would have the potential of being a way to delay or slow the progression of Alzheimer's disease. And we talked about just one other brief mention about a great deal of interest right now in risk reduction therapies that are based upon altering lifestyle. Our lifestyle, if you hadn't noticed, is horrible. We don't exercise enough. We eat too many fats, and we don't treat hypertension or diabetes properly. We gain weight and develop these metabolic syndromes that are all risk factors for Alzheimer's disease. A study that was done of populations in the UK of this generation versus the pre-war generation showed that the incidence of Alzheimer's has significantly decreased in the younger generation who has a better diet, who has better medical care, treating hypertension, going to the gym, etc. You can imagine pre-war what people had to deal with, and that lifestyle was contributing to the risk, we think. So there's a big push to accelerate that kind of approach in addition to the drug treatments. And so I'll leave you with this. because exercise is such an important element, and it's been validated now over the past several years. And every time I see this, I feel guilty that I have to leave and go to the gym immediately but I'll leave it at there, and I think we're going to have another presentation and some questions and answers. Thank you.
Thank you so much. So I invite all the panelists to the stage, and I have the pleasure to ask the first round of questions. So, but actually you have already answered any kind of question I could ask, so I have to come up with some new ones. I have a very basic, very simple question that has nothing to do with research, but if autophagy is so important and such an integral part of the cell's function, and there has been research for so long, why is there so little in normal biochemistry books or in any kind of school book? There is only just a bit in it. Do you know?
Well, it was not actually discovered until the 1960s. Christian Dedoof at Rockefeller University won the Nobel Prize for describing the phenomenon, and there was a certain amount of activity for about a decade, and then there was this silent period for no good reason for at least 3 or 4 decades, and then the turn of the century, people started to realize this was very important, and that's what's. And the second Nobel Prize was awarded 2 years ago for the mechanisms of autophagy.
Yeah.
Pretty recent described phenomenon.
So just to say it again, what exactly is the role of autophagy in Alzheimer's disease? I mean, you elaborated already, but to just be able to grab it again.
Well, as pointed out, there is disturbance of this pathway in the neurons, and you have to realize that neurons are cells that are not dividing anymore as compared to other tissue in your body. So, our neurons, they stay all of our life, and they have to take care of, clearance of junk and all these functions all the time, while having a lot of impacts from different risk factors. Ralph mentioned some of them. So there's a huge demand of clearance of defective proteins and organelles, and this obviously, as pointed out, is disturbed, and this leads to clumping of neurons, which means there's no longer a proper trafficking going on in the neurons, because neurons are quite large cells. If you consider the ischiatic nerve from being around 1 meter in length, you can imagine what the transportation along this nerve really, how demanding this is. So if there is clumps there based on the defective autophagy, it's just not working anymore. And this, of course, is going on in the brain and leading then to dysfunction.
Thank you. So you already mentioned it, but you would see autophagy more as a target for prevention and stabilization versus a treatment or a cure? Or both?
Well, a cure in the context that I described, the train analogy, is really unlikely, because by that point, you have so much cell loss, and the brain cannot regenerate very effectively. There's a little caveat about that, but for the most part, you lose your neurons, they don't come back. And so trying to cure something that involves having more neurons and more connections is not realistic. So the goal is really to prevent, that would be the ideal, but to intervene, which is sort of a compromise where you have minimal brain cell loss, but you have some of the markers that would indicate an early stage of the disease. That's why biomarkers like the imaging and blood markers are such an important thing, that we're looking for that sign of the early stage that we can then apply these drugs, and that is more likely to be able to slow the disease. But you know, we talked a little bit today about lifestyle as a way to actually delay the onset of Alzheimer's disease, and that's really within our ability to do, is to adopt lifestyle changes, diet and medical surveillance and exercise that have been shown to delay the onset. This is how it's been evaluated, that they're effective measures, is because those people have a later onset or no onset of Alzheimer's.
And so what can we learn from Alzheimer's related diseases in this context, and what are the causes of these types of neurodegeneration?
Well, there is a hope, and we discussed this also during the day, that some of these neurodegenerative diseases, which are mainly age-related, they have common pathways, and we were talking about diseases, at least in part, Parkinson's, but mainly also on amyotrophic lateral sclerosis, as compared to Alzheimer's, all showing a disturbance in the protein clearance, so accumulation of protein. So why is it's not cleared, and why is the system no longer functioning? But we can learn from related disease, perhaps also with respect to Alzheimer prevention or intervention, early intervention, when we look at very defined diseases. So and we have discussed one example, this BPAN. I was mentioning it shortly. So perhaps for those in the audience that do not know this disease, this monogenetic disease, you may give a short introduction to it and what it really means.
Yes, sure, I can try. For this, I have to state I am a representative of a patient organization from Germany, and we are representing patients, it's mostly children, who actually have that disease. BPAN, this is caused by a mutation of one gene on the X chromosome, and this gene codes one of the proteins that are important for the early parts of the autophagy running in the body. And one main symptom for the clinical diagnosis, this is important, is that it accumulates iron in the midbrain. So, but the role of that iron is not very much cleared up to now. But still, this is autophagy basically that is not running properly, and this produces symptoms in children already at very early age, which first presents as a development disorder. Then often, almost all children, they have different forms of epilepsy, and most of the children, they do not develop any speech, any language. Some of them, they can say a few words. Generally, communication is very problematic. Not even sign language is something that at least many of these children can actually learn. And I must say, I can also describe it from my own perspective because my daughter is also affected by this disease. She's now 7 years old. She goes to school, a special school. But for us, that means the life that we lead is very much different to the life to many other families. So our children is dependent 24 hours on us, on our help. She cannot run, she cannot move, but we know that she recognizes much more than actually she can express, and this is something that we actually cherish, and well, it's a difficult situation, but we have to learn with that.
Thank you. So, where would you see the potential of autophagy in targeting diseases such as this and targeting genetically caused diseases versus non-genetically caused neurodegeneration for the whole?
Well, I can try. Well, I've discussed this matter already with one of the scientists here because, and you said it, at a late stage when we see already the symptoms, the cure of the disease is basically impossible. So that means if you want to attack this disease also with this, say, genetic alteration, we have to start at a very early age. That means we need some kind of screening already with newborns to actually find out if they have this genetic mutation. Then maybe there's a hope that with a genetic treatment that basically the symptoms maybe do not even show up or at a very mild stage. For us at the moment, the most important thing that we could try, or this is the main goal, is to chronify the disease. That means you have to know this is not how it stays. This is a degenerative disease. From all what we know is that at a certain age, mostly people say at young adulthood, these children, they degrade heavily, very quickly, with strong dementia, with strong dystonia, which means she will be bedridden, she will probably not recognize us anymore, and typically many of the children, they die at very early age. So that means if we have the means to avoid that degradation, that would already be very helpful. And at that point, we have to look into the autophagy and see if there's a way to somehow modify what is actually going wrong. And I think there are some ideas going on. There are some ideas on the market, but of course, the research is only beginning right now.
Yeah, you're right.
But what this also tell, if you try for a second to be, to take away the personal empathy, but as a scientist, what does this really prove? It proves that a dysfunctional autophagy is causing dementia, in this case, a developmental type of dementia, yes, which then is also a big hint and a big motivation to further go into detail in this process also in other neurodegenerative disorders. So that's sort of a lifetime proof that if this process is really disturbed, it can lead to this devastating dementia phenotype. So for this, it can act, and this was the initial question, what can we learn from rare disease for the big ones, number-wise, so to say. Every individual is it's a horror for every individual. But if you count on numbers, okay, of course, we need new ideas for the prevention. And this could be one hint. Say, well, let's try to stabilize autophagic function in neurons, and then this may be one way to prevent, but not to sound too optimistic at this point in time. we are very early in the stage of knowledge how this, as we learned today during the workshop, how this is really going on, and it's not as clear-cut as in the BPAN case, basically.
Yeah, and from my perspective, so at the ADDF, our primary goal is to find drugs and treatments and put them through clinical trials so we can get them to patients. And we scan the field, we look at the science, and we see what areas are new and emerging that could be targeted for drugs. And these rare disorders give us a sort of proof of concept that show, these really could be involved in the disease and help us identify targets that we can start to develop drugs and therapeutics to really go after fixing these problems.
So you do a lot of networking as well, right? So you have connections to other associations. You don't only target Alzheimer's disease, but other dementias, and you work with other associations to
Sure. So I think it was mentioned already, but we have a lot to learn from other neurodegenerative diseases and also other diseases outside of the neurospace, including hypertension and diabetes. There are a lot of chronic conditions that have been comorbidities linked to Alzheimer's disease. But in the neurodegenerative space, we have a lot to learn from the Parkinson's field, from multiple sclerosis. Inflammation is a major contributor to Alzheimer's disease, and there's been some really great progress with the biomarkers, a biomarker of neurodegeneration that's been more validated in multiple sclerosis that we can try to adopt in the Alzheimer's field, as well as therapeutic interventions. And so at the ADDF, we really try to learn from, what's going on in other conditions and see where the successes are and try to follow in those footsteps.
So we heard today that there are a lot of compounds out there actually that are already being screened that are good for targeting autophagy, enhancing autophagy, but we still say we're far away from really grasping where to go with them or where to really apply. Where are the complications actually? So why are we so far away from a real translational step?
Well, with any new avenue of investigation of a therapy, the time frame is a lot longer than you might imagine to do the basic research and to actually validate it in models, cell models, mouse models, is 4 or 5 years of effort, and then the clinical trials. The phase 1 is one where a lot of drugs drop out because they have side effects, and so in a field like autophagy that really didn't. Our paper in 2005 was the first to really make the link that autophagy was very abnormal in Alzheimer's disease. And so what's that, 14 years? It took another 5 years for people to say, oh, maybe he's right, there's something going on here. So it's been 10 years. So you can see the time frame for actually doing something that's meaningful has been very short, but it's exponential, and having a meeting like this is indicative of the fact that we've created a critical mass of people, and that mass is going to be even double or triple next year or the following year. And that's how the acceleration occurs. So it starts slowly, but then it builds. So that's where we stand. A lot of things are being tested, but that testing takes a while.
Add one more thing. One of the most critical components of developing a drug is having a biomarker, and you can see whether the drug is actually working and whether it's hitting its target, and you can monitor that in human patients. And so in addition to drugs, we also need to focus on developing new biomarkers so they can measure disrupted autophagy in human patients and then figure out whether any intervention is actually working.
And I think this shows how important it is to get connected, to get the input from different research areas, especially in neurodegeneration, and that's what we had today. A lot of people who work in autophagy, but different neurodegenerative diseases actually came together, which is not very common, and exchanged and connected, and I think we had a very constructive day today, and I think it's a great step in the right direction. And this is my last question before we open the Q&A session. So what is the biggest impulse you take home today from this day of exchange?
The biggest impulse, well, we have learned that we created lots and lots of data, and good data, and reproducible data, and we have learned about the significance of this process, but we have also learned that autophagy is a very basic process, so it's hard to fiddle around, pharmaceutical with compounds with a very basic process, which is involved in disease, but which is also involved in your basic physiology of the cell. Nevertheless, what I have the hope is that indeed with this new knowledge in particular in neurons, so we started in the autophagy research in yeast, now we are already in the neurons, which is completely different from yeast, but the basic mechanisms are sort of the same. So I think it's a very, very good step forward that we are now in the cell type, which is really relevant for diseases such as Alzheimer's disease. So I'm very optimistic that this drives research further and gives us also motivation to do even more work based also on the experience that we hear from families that are affected by diseases.
Right. Well, my impressions are basically twofold. The first thing is, by the sheer complex of the entire autophagy system, I'm a little bit overwhelmed, I must say, because my point of view was more or less restricted to this disease that I'm trying to work out with lots of help from different scientists and clinicians. So it's getting even more complicated. I must say I'm a scientist myself, but I'm not within biochemistry or so on. So it's a very different realm. The other thing is, because I come from the side of applications of all this research, is that it seems to me there is already a lot of fundamental research going on, and I think my impression was that people are growingly getting to understand what's happening there, and basically the main processes, some of the main processes are already very well known, I guess, what I discovered today. But when it comes to translate that into applications, there's a very different thing. You get into regulations. How do you actually measure something? How do you measure actually that you improve a disease? This is very difficult. And I think this also needs a lot of work, and you need your different stages. If you want to do that, you do first do, it's like that. You have to face it. Animal experiments with mice mostly, but then later on you sometimes also have to use primates. This is how it is. And until you get something like a clinical trial, it takes years, and then in the end, very often you are disappointed because it doesn't work. So this is very difficult.
I think what this day did was to persuade our colleagues working on different diseases that there's really incredible value in coming together to talk about the commonalities and the differences, focusing on autophagy, but in relation to different diseases. And you would think this would be an obvious thing to happen, but this was basically, I think, the first meeting that I've seen that brought people that are working on Huntington's disease, ALS, Parkinson's, Alzheimer's disease. All of these diseases have their own meetings, and they may talk about autophagy there, but they never see or even meet someone who's working on Alzheimer's or some other disease. And I think it opened people's eyes as to how this can really accelerate research by combining our collective wisdom on these different diseases.
Thank you. For me, the thing that I took away from today, first of all, is the incredible science that's going on in this field. These scientists that presented today are doing hard work trying to figure out a very complex process that is autophagy. And it's been really great seeing all of the progress that's being made in understanding this system. The second thing that I took away from today was just this gaining momentum in the field. And, you know, from my perspective and the drug discovery perspective, we're constantly looking for things that we can develop drugs for. And autophagy is an incredibly exciting area, but just still understanding what is going to be druggable, if anything, and how to do that. And so for me today, seeing this building momentum and this gathering of scientists that are really moving in that direction and pushing it forward has really been inspiring for me.
Thank you so much. And hopefully we will have many more meetings like this that will push us forward. With this, I close my question session, and we're happy to take questions from the audience. If you have any.
Thank you very much. That was very elucidating. And I'm a clinician, a doctor, and it was a very nice discussion for someone who doesn't really get exposed to that pathophysiology, so I appreciate it. Question I have several, but I'll get to the main one. Is it the chicken or the egg? Is it the amyloid and these tau particles that disrupt autophagy, or are they the result of the autophagy system being dysfunctional?
Well, there was actually a talk by one of the participants who actually said that it's both. And this is the case, I think, with a lot of these proteins, including amyloid-like proteins, is that they disrupt the very process that would be clearing them, and that promotes more accumulation. So it's both the chicken and the egg, I guess. And this seems to be a common theme that one promotes the other to happen.
If I can follow up very quickly,
Yeah.
On the autism spectrum, has anybody ever looked at this in all these kids that have autism? Is there a dysfunction in their way the autophagy system works in neurons?
Well, with BPAN, this is certainly the case. The autism is also a typical symptom with BPAN and other diseases in this MBA spectrum where at least iron is deposited in the midbrain.
Yeah, at the very molecular level, synaptic level, the communication between the cells, there's remarkable similarities in the machinery that is disrupted. There are different disruptions of that machinery, but there is understanding how the machinery works is going to have information that's relevant to autism and the cognition problems in that realm and those in other diseases like Alzheimer's.
Any other questions in the audience? Oh yeah, I'm coming.
To what extent is funding adequate for the research, and if not, how much is that delaying progress on the research?
I can speak for the U.S., which it was a huge problem, and this was until about 4 years ago, and for a period of 15 or so years, there was flat funding at a very low level for Alzheimer's disease, $400 million, which compared to cancer in the $5 to $6 billion range. And there was an advocacy spearheaded by the Alzheimer's Association, among other advocacy groups, to push Congress to accelerate the funding, and now the funding is up to $2 billion. So this is a real miracle in terms of accelerating, but it's been only 4 years, and some people dropped out of the field because they couldn't get grants. Now they're coming back in, or we're training new people, so it's a ramp up. But it's very promising that now NIH has stepped up. I don't know, from the German side, it's always been pretty good.
Yeah, it's pretty good, and of course, if you are asked as a scientist, it could be always more and more and more. But I think, in particular with Alzheimer's disease, it's frequently not the lack of money, but the lack of new ideas. And I didn't want to use this podium now to extend in more and more my personal opinion, but even if we hear from the audience, again, the amyloid thing, you have to realize that actually it is Alois Alzheimer's fault that we are talking about amyloid and tau. He looked through the microscope and saw it. But if you read the initial report from 1907, he also reports on other things like vessel changes. Okay, we know today that cardiovascular risk factors, hypertension, and we overlooked mentioning of what he called lipid sacchels, meaning aggregations of lipids. Why did we overlook this? Because in science it's also a matter of methods. At this time when the protein Alzheimer research took off, there were protein methods ongoing, but lipid methods were not very well developed. And my personal opinion is that for some cases, the amyloid plaques and tangles, they might, be the ultimate cause of the disease. But I consider Alzheimer's as a wide range of different diseases, which finally comes up with the dementia and with some common pathology, but the initial cause is based on the personal history, decades and decades that you live before you show signs and symptoms. So I would make a strong point in, that's actually I stopped Alzheimer's research, the pure Alzheimer's research, when I realized that I don't get any grants if I'm not mentioning amyloid. So I said, well, okay, so let's forget about it. So we have to open the box, think outside the box, and not gather weird ideas, I mean really basic science, natural science-based new developments. And my hope is that what we did today, the autophagy part, is one step in this direction. And again, I don't want to say that the amyloid has nothing to do with the disease, but there might be a particular fraction of patients. And knowing that we do, amyloid is one of the best understood, biochemically best understood molecule in molecular medicine. And it is around concentrated research about 30 years. I actually started my career also with amyloid on the West Coast in the 90s. So, but there's nothing that we have in our hands now. And even if the newspapers are now full of the reinvention of whatever immune trials, it appears weird to me that a huge company drops out a drug and then after 3 or 4 months says, well, we started to recalculate, we have a few more patients, and that's why I'm a little bit skeptical if this is really the right way to go. For some patients, yes, I really repeat it again, but we have to open up and maybe realize that it's Alzheimer's diseases rather than one entity. But
Can I quickly add a little aspect to that? Because you were specifically asking about funding situation. It's always depending on the actual disease. When you're talking about these widespread diseases like Alzheimer and Parkinson's, I think the situation is pretty okay. But most of the neurodegenerative diseases, they are also the rare diseases. And they share the same fate like all the rare diseases here. The funding is really poor. Yeah? So public funding is really poor. As far as I know, there's really not one program in Germany for public funding like that for rare diseases. There's a little bit from the European Union, but apart from that, it's really, it's bleak.
My name is Howard. I'm not from this field, so I'm an outsider. I have some simple question for Dr. Nixon about the brain inactivity and the low education. And based on your chart, people at 40 should start exercise, start prevent Alzheimer's disease. So that comes to a question, its brain is not a muscle. So muscle you can jump and run and exercise. How you exercise the brain, and whether music, color, painting could be a good avenue, or even rock and roll, loud noise, or quiet meditation.
Well, there, in fact, are a growing number of studies. One of the offshoots of having a bit more money is that we can explore avenues that include Eastern as well as Western approaches to therapy. And as you know, you probably felt better after you had this treatment, which indicates what we already know, which is that when you feel better, your brain is infused with hormones and growth factors and various things that give you resilience to disease, improve your immune system, etc. These are all factors that may play a role in reducing risk. So it's not, the brain isn't a muscle, so you're not contracting it and moving it around, but it is being constantly bombarded with factors that are both good factors, therapeutic, and toxic factors. This is why things like acupuncture, massage, relaxation therapies, but also stimulating your brain. This is one of the things that has evidence base now, is that education, which implies that you later in life continue to read and to communicate with people, these are things that activate hormones and growth factors that are very beneficial to your brain.
Thank you. One last question was here, I think.
Yeah, just I work with wounds, burn wounds, and a lot of the research done on that showed that apoptosis and autophagy worked in tandem. So I was wondering whether the decrease in this brain mass that you see, is that apoptosis that's occurring as a result of a defect in autophagy? But you see actually shrinkage in the brain mass that you showed, and I was wondering if that was sort of working in tandem with autophagy, that you would see an increase in apoptosis.
Well, there is a particular pathway which is a autophagy-dependent apoptosis, which is highly related to cancer cells actually, and is defective in cancer cells. But what we see in the Alzheimer's brain is I would, from what I know and from the reading and from the paperwork and from the basis, it's apoptosis going on, and not so much apoptosis in the first place, you know, taking away whole tissue, but rather you have a degeneration of the axons and you know, and then finally remove, you know, the cell disappears by apoptosis, which is, as you know, a non-inflammatory process. So that's, you know, occurring all over ages, not over ages, but over decades. So it's hard to follow it it's hard to simplify it in a model system actually. There's even, in mice, there's no good models showing where you can induce a real neurodegeneration. A few of them are now available, but it's not like that you pump a mouse full of amyloid protein, then the brain falls apart. It's not like that. So it has to take a lot of time before the cells then finally goes into apoptosis. But coming back to your question, I don't see this.
It's not working in tandem.
No.
Okay.
Fuzzy,
there's one more? Okay.
This should be the final.
One last question.
Has anyone ever studied small groups of people living in isolated areas where they live over 100, and the population in general lives to very old age? Why don't they get Alzheimer's, or do they? Are their brains different? What's going on there? And that should teach us a great deal.
They do get Alzheimer's, but they are more resilient for the same reason that they are resilient to the effects of aging, the onslaughts of aging. And there are populations like in Okinawa and different parts of Japan where, but elsewhere as well. And usually, the answer is that no one really knows all of the factors, but part of it is genetics, that this is a population that is fairly closed, and if they have good genes, they're the ones who are going to perpetuate those because they stay in the same location. But even more importantly, it seems as though diet and a healthy lifestyle is very important to longevity. And I would just add that good autophagy is one of the most important factors in determining lifespan in not just humans, but in other species. So if you clear proteins well, you might live longer. So that's maybe where we should leave, the best message we could leave you with right now.
Thank you so much. Well, with this, we will wrap up the evening session. We would like to thank the German Center for Research and Innovation for co-organizing the event with us. The team has done a wonderful job in supporting us. Thank you so much. Thank you to the scientific organizers, Professor Ralph Nixon, Professor Christian Bale, to putting together a great program, to all the scientists who came together here to exchange knowledge, to our other panelists, Dr. Mariel Owen and Dr. Marcus Nielbach, and of course the audience. Thank you so much. We had a great day, we had a great evening, and now there is a small reception. Thank you.